Hello, everyone. My name is Alisher Talgatov. I'm a graduate student at UT's Department of Chemistry and Biochemistry, and today I want to present the research that we do at Dr. McFarland's research group called a poster called Applications of Photoactivated Metal Drugs for Cancer Therapy. PDT, or photodynamic therapy, is an alternative strategy towards the cancer treatment, and it employs the drug called photosensitizer. And once it's absorbed for a certain amount of time within a tumor, it's being exposed to light, and then light causes reactive oxygen species to kill cancer cells. And recently, metal drugs are found to be interesting because of their tunability and accessibility to the different excited states. And our own ruthenium-based drug called TLD-4033 is currently undergoing phase 2 clinical trials against non-muscle invasive bladder cancer. And the objective of this work is to synthesize and characterize and test the new class of ruthenium polypyridyl oligotyphenyl compounds, thionyl compounds, and determine the effect of the oligotyphene chain on the identity and the biological activity. So we synthesized the family of 7 compounds. Synthesis involved the microwave irradiation at 180 C for 15 minutes, and crude products were isolated as hexafluorophosphate salts and purified using flash chromatography on silica, followed by the conversion to their corresponding chloride salt on the anion exchange column amber light. And finally, we purified our chloride salts using cyclic chromatography on separates. All 7 compounds were characterized by 1D and 2D NMR techniques, mass spectrometry, and purity was confirmed by HPLC. And later, we evaluated photophysical properties of our compounds. This is the UV-Vis spectral overlay of all 7 compounds, and here we can outline 3 major features, where IL stands for interligand transition within coligands, IL/ILCT stands for the intraligand or intraligand charge transfer within the imidazophenanthroline thiophenene ligands, and with the increasing of the number of thiophenes, intensity is being increased and the peak is more redshifted. While for the MOCT region, or metal to ligand charge transfer, we observed that the intensity is also increasing, but the peak stays on the same value of the nanom in terms of wavelength. We also performed differential spectroscopy of the excited state absorption properties with a technique called transient absorption. And for the ruthenium without any thiophenes, we observed the bleaching around 450 nanometers, which stands for the depopulation of the MLCT state, while for the complex with the 4 thiophenes, we observed not only depopulation, but also the population of the triplet ILCT state, which is much more longer lived. And later, we confirmed that this longer lived excited state was the crucial for the photobiological activity, where testing against the SKML28 skin melanoma cancer cells, we observed the ruthenium-40 was the most active, as expected, not only on normoxia, but also in hypoxia. And to conclude this work, we synthesized, characterized, and tested new family of compounds against cancer cells. And to achieve higher biological activity, we need to get to the triplet ILCT state. And how to get it upon photoexcitation, we have to increase the number of thiophenes within our molecule to get the energetically favorable positions to access this state. We want to thank our funding agencies as NSF and NIH. We want to thank UTA, Department of Chemistry and Biochemistry, and UTA's Department College of Science for organizing the Discover Symposium. And if you have any questions, reach out to our website and social media. And thank you for your attention.